No, think of the place where exchange takes place. So think of the alveoli. That's the respiratory zone. Sometimes it seems like they include those terminal bronchioles in there because that feeds in to the respiratory zone, but it's the alveoli where exchange takes place. That's the zone. Okay? Any other questions so far? We good? All right, so let's get going with pressure. We know that we need to create a pressure differential in order for us to breathe in. To breathe in for gas exchange, for all that good stuff. So we ended up talking about inspiration and expiration and some of the different muscle groups that were going to help me do that. Remember that? Okay, why do we breathe in? Talk about physically why we breathe in. What do we do to the thoracic cavity? We make it larger, we make it bigger. How? Diaphragm moves down. What do we call that? Be scientific. Moves inferiorly, kind of spreads things out. And who else is going to help pull up on those ribs? That's for a deep breath. For a regular tidal volume, who are the guys that are going to help pull on the ribs? Intercostal muscles. So that it's going to act like kind of like almost like opening an umbrella so that I can inspire, bring air in. And then I'm going to do the opposite for expiration. So know those muscles and what goes on. So in this diagram, we're talking about intrapulmonary. What's that mean? Pressure inside the lungs decreases as lung volume increases during what? Inspiration. Pressure increases during expiration. Everybody understand that? Whose law? Boyle's law. And what's Boyle's law? Pressure is inversely proportional to the size of the container. Intrapleural. Plural. What's that? What's the plural? Yeah, those are the membranes that line the thoracic cavity. So those are the things, what kind of membranes are those again? They're serous membranes. They secrete a fluid. So that pressure is also going to be important for inspiration and expiration. So intrapleural pressure, pleural cavity pressure becomes more negative as chest wall expands during inspiration and returns to the initial value as the chest wall recoils. It's kind of like an elastic. When I pull it to stretch it to make it bigger, then I kind of let it go, it's going to snap back to where it originally wants to be. And that's what recoil is. And then volume of breath. During each breath, the pressure gradients move 0.5 liters of air into and out of the lungs. Give me another name for that. That volume of air. That's my tidal volume. Okay? So that, you should understand that diagram and relationship of pressures discussed in that diagram. So, physical factors influencing pulmonary ventilation. Of course, these guys, the differences in pressure, whether it be in the lungs or in the pleural cavity itself. Resistance, is that going to affect pulmonary ventilation? Yeah, and what's resistance? Yeah, it's kind of me scraping up against the sides of the tubes. Now, if I have problems with the diameter of the tubes or not being able to change the diameter of the tubes, that is also going to affect airway resistance. So the diameter of conducting zone is a big factor in airway resistance. As long as I can get air through those tubes without too much resistance, I can get air to where? The important part. Somebody asked a question about that just before class started. What's it called? The respiratory zone, where I need to do my business end of gas exchange. So that's very, very important. We talked a little bit about the respiratory zone and the cells that were hanging out down there, type 1 alveolar cells. We talked about surfactant cells. And surfactant cells are going to play a large role in decreasing surface tension, because surface tension is also going to play a role in gas exchange. Everybody understand what the surface tension thing is all about? The higher the surface tension, the harder it is for gases to do what? Exchange. You say exchange, be more specific. Go from gas into liquid, where they have to then be transported through the system by who? What kind of liquid? Blood plasma, and who else is going to help carry respiratory gases as well? Besides the plasma, who else is going to play a big role in that? Who? Red blood cells. Specifically, what was that molecule called? Hemoglobin. So surface tension is also going to play a role in pulmonary ventilation. And surfactant is going to help decrease surface tension to help with gas exchange. Don't forget to read up on homeostatic imbalances on page 823 and the discussion about infant respiratory distress syndrome, which is related to surfactant production. The other thing that is going to affect all of this movement is compliance. Compliance. What's that? Yeah. In an unfancy scientific way, it's kind of the stretchability of lungs. Remember, I have to be able to inflate those balloons to fill them up with atmospheric gases so I can have gas exchange. So lung compliance is also a big factor in moving air to the respiratory zone. If I can't blow up those balloons, if compliance is decreased, I'm going to have a hard time getting enough air for gas exchange. Okay? So that was like a little bit of a review from last class. What's this diagram of? Increased resistance in the conducting zone. Where's the highest resistance in the respiratory system? In the medium-sized bronchi. And we talked about the effects of things like asthma and emphysema, or anything maybe related to a chronic obstructive pulmonary disease and the effects on those particular airways and resistance. So we have least resistance in the terminal bronchioles dumping into what? The alveoli, or the respiratory zone. I'm not going to go through these again because you hopefully know them by now after doing your lab homework for this week. So, respiratory volumes, know them. Tidal volume, inspiratory reserve, expiratory reserve, residual volume. Be able to calculate some of the lung capacity volumes, inspiratory capacity, functional residual, vital capacity, and total lung capacity. Again, we talked about all of this good stuff last week. Dead space. Dead space is what? Anatomical dead space. We kind of ended up talking about this. Yeah, it is proportional to your weight. Yeah, it's something, it's the air that doesn't make it where? To the respiratory zone. Okay? And it is related to weight. So the higher my weight, the less what? Less of my tidal volume gets down to where? The respiratory zone. So rule of thumb is that the anatomical dead space volume in a healthy young adult is equal to 1 milliliter per pound of ideal body weight. So what's going to happen to dead space when I maybe have a problem with compliance, or maybe I have a problem with resistance? Or maybe I have a Twinkie issue. Less in the respiratory zone, more in the dead space. Exactly. Pulmonary function tests. They talk about spirometers. We used a very high-tech one in lab, which you all were able to measure volumes with. So I'm not going to go into that too much because hopefully, like I said, you kind of cleared that up when you did your lab homework. And that is talked about on page 825 and 826. Non-respiratory air movements. Coughing, sneezing, crying, laughing, hiccuping, and yawning all involve the respiratory system, but we don't do them because of respiration. They're going to help clean things out. Like, why do I cough? Yeah, get stuff out that maybe came in. Sneeze, same thing. Crying, well, I'm Italian, so I know all about that. Laughing, something's funny. Hiccups, why do I hiccup? Yeah, you kind of have a little spasm in your diaphragm. Why? We're not quite sure. And yawning, yeah, that's what they say. You yawn because you need to bring in more oxygen to hopefully get your brain cells working a little bit better and not falling to sleep while we're talking. I should videotape those of you who fall asleep in class. It would be good. It would be good. Try not to keep your mouth open though when you fall asleep, because you don't want to drool on yourself. So, gas exchange between blood, lungs, and tissue is what we're going to talk about next. Now, we talked about pressure. We talked about Boyle. Well, let's talk about Dalton, because Dalton also plays a role, and Dalton's theories also play a role in gas exchange. We know there's gas in our atmosphere, right? Where was that little chart? There it is. So, what is the gas in our atmosphere? Believe it or not, most of it is something we don't even use, at least with respect to the respiratory system. Most of our atmosphere is what? Nitrogen. The next in line is oxygen. So most of our atmosphere, almost 99% of our atmosphere, is nitrogen and oxygen. Well, the oxygen we use, the nitrogen we use, but we can't get it in through the circulatory system. Somebody else has to do that work for me. Do you know who's going to do that work for me? Hopefully you eat them every day. I bet you some of you don't. We get a lot of our nitrogen in proteins, but we actually use something from somebody else to make those proteins. Vegetables, fruit, plants. Plants have the ability to fix nitrogen, so we're going to use a lot of the nitrogen from plants to help make our proteins, and our proteins is where we get a lot of our nitrogen from. So, carbon dioxide and water vapor is also part of our atmosphere as well. Now, think of your atmosphere as a whole bunch of these guys banging around in this classroom. That's your atmosphere. Molecules are in motion, correct? So you have all of those guys banging around in this, pretend it's a beaker. Now, the speed at which they bang around or exert their pressure is not all the same. It depends on my size. So each of these guys, nitrogen, oxygen, carbon dioxide, exert their own pressure on a container. Am I making sense so far? So say you were all in this room and I told you to run as fast as you could and bang against the wall and keep doing that. Would everybody be running at the same speed? No, some of you are zippier than others. Some of you are a little slow. Same with these guys. So the speed at which they bang against their container is what we call their partial pressure. And the sum of everybody in the room, or everyone in the atmosphere, is going to exert a atmospheric pressure. Everybody with me? So your partial pressure is the individual pressure you contribute to the total. So nitrogen's partial pressure is what? 569 millimeters of mercury in our atmosphere. Remember, our atmosphere is kind of like a petri dish too. So when we generate things in our atmosphere, what happens to the things that we generate? This is kind of an ecology plug. They go out into the atmosphere and they bounce around in this closed atmosphere. Now, some things are good at picking up stuff that maybe we create excess of. Anybody know where I'm leading? Remember those plant friends I was telling you about? Yeah, when we generate too much carbon dioxide, our friends, the plants, actually use carbon dioxide. So they're going to take some of that out of the atmosphere, and they're going to be nice to us and generate what for us? Oxygen. That's a waste product for them. What happens when we generate too much carbon dioxide? Can the plants keep up? No. What's that cause? Yeah, our carbon footprint may lead to an increase in carbon dioxide in our atmosphere. What do they call that? Global warming. Is that real? Yes, it is. That's my ecology plug. No, it's not a political move. It's real. And every time you get in your car, you create more carbon dioxide. Every time you breathe out, you create more carbon dioxide. Yes? So that's real. That's not a political move. And the more carbon dioxide in this atmosphere, the more pressure is created in this atmosphere. Again, so we have to kind of even things out with maybe being a little more ecologically sound. And what's going on over in Japan? Yeah, correct. Lots of problems, lots of things on fire. Guess what's being released into the atmosphere? This is a petri dish, my friends. So will that affect you? Yes.
So it says like there's 100 people exposed to radiation, but it's going to gather in the atmosphere, right?
Correct.
And it's a form of cloud.
Well, just like think about it, and not to dilute it at all, which is what's going to happen, but it's going to disperse more in the atmosphere. So eventually, it's not going to be as harmful as the concentrated amount that's dropping on people right there. Not to be disgusting or anything, but it's like pissing in the ocean, right? Well, that's a good visual, right? But, and again, not to be too disgusting, if you keep pissing in the ocean, eventually the dilution factor is going to go down, yes? Or throwing this stuff into the atmosphere. You do the math. So we have to start thinking about things like this. All right, back to the atmosphere, back to anatomy and physiology. That was my plug for the planet. So nitrogen 569 is the pressure it exerts in our atmosphere. Oxygen 104, carbon dioxide 40, water vapor, or water in gaseous form 47, for a total of what? 760 millimeters of mercury. Yes? And what's that? That's our atmospheric pressure. So does everybody understand how all of the stuff in our atmosphere contributes to that number? Now that's the number where? If I go up Mount Everest, is that the number? No. What happens to this? It goes down. So it gets lower as I come away from what? Sea level. So at sea level, when I change my position on this planet, I actually can change this. As I go away from sea level, up in the sky, if I go up in a plane, for example, what do I have to do in that plane in order for me to be able to keep breathing? I have to pressurize the cabin. How come? Right, because remember, the reason I breathe in and out is because I can create a container with lower pressure than that, so the air will come in. But if I go up in the plane and I don't pressurize my cabin, can I do that? I physically cannot make my lungs, or thoracic cavity, I should say, that large. So the air's like, well, hell, I'm not going in there. I'm only going to go from a high pressure to a low pressure. The law of partial pressures is Dalton's law. So don't get the two guys confused. Dalton's law of partial pressures states that the total atmospheric pressure is a result of the culmination of the partial pressures of the gases in that given atmosphere. Yes? No. Dalton says, and let me see, states that the total pressure exerted by a mixture of gases is the sum of the pressures exerted independently by each gas in the mixture. And that's the first sentence on page 827 under Dalton's law of partial pressures. Okay? So you understand what he means? Okay. And then Henry, of course, he had to have his 2 cents in the respiratory system. What's Henry's law? Well, Henry says when a gas is in contact with a liquid, the gas will dissolve in the liquid in proportion to its partial pressure. You see how all these guys kind of relate to each other? So I'm only going to dissolve in a liquid if the relationship between my partial pressure and the liquid's pressure that's being exerted, say of oxygen, is a gradient in which I go from what to what? High to low. So always remember, I'm going to go from high to low no matter what. That's where I want to go. So if I want to go from high to low, I have to create that pressure difference so that because of Boyle and Dalton and Henry's theories, that's how gas exchange will take place. Are you with me? Okay. So this diagram is a nice diagram because this diagram shows us some of the differences and the reason why gas exchange takes place in the system. When I suck in air, oxygen, now we're just talking oxygen here. Remember, what was total atmospheric pressure? 760, a total of everybody. But oxygen is exerting a partial pressure when I breathe in of what? Look at that diagram. Don't look at this one. 160 millimeters of mercury. Why is it at a higher pressure now? Because I'm sucking it into what? Yeah, exactly. I'm sucking it through a tube. I'm creating a higher pressure. Even though, and I knew you were going to say this, but it says right here that the partial pressure of oxygen is 104 millimeters of mercury. Yes, but now you're sucking it through a tube. You're creating a suction. So now when you're inspiring that air, it's exerting a partial pressure of 160 millimeters of mercury. You with me? I didn't see any heads go. Yes? How about carbon dioxide? 0.3 millimeters of mercury. So I suck in that air, gas exchange is going to take place, alveoli of the lungs. What's the partial pressure of alveoli? Oxygen in the alveoli of the lungs? 104. Oh, where's oxygen going to go? From a high pressure in the inspired air to the lower pressure in the alveoli. Well, how about carbon dioxide? Where's that going to want to go? Holy moly, 40 millimeters of mercury in the alveoli. Where am I going to go fast? You see? Okay. So in the pulmonary veins, P. The P means what? Partial pressure of oxygen is 100 millimeters of mercury. When blood leaves the lungs, entering the tissue capillaries, you see again the difference in pressure. When we get to the tissue level, we see again a difference in pressure. You seeing this? So oxygen in the blood, 100 millimeters of mercury. In the tissues, what's it exerting in the tissues? Less than 40 millimeters of mercury. Where is that oxygen from the blood going to want to go? From high to low. Do you see why we get exchange at the tissue level? Again, difference in CO2. It's higher in the tissues than it is in the blood. Where is it going to want to go? To blood. And where is blood then going to bring it? It's going to bring it to the lungs, and we're going to start the whole spiel over again. Yes? So the differences in partial pressure are also going to play a factor in gas exchange. Yes? So this is a nice diagram because you can visually see those differences. What's this a diagram of? The oxygenation of blood in the pulmonary capillaries at rest. Where are the pulmonary capillaries? Yep, in the alveoli. So, in your textbook, they talk about composition of alveolar gas, external respiration, partial pressure gradients and gas solubilities, and all that is fancy stuff for what that previous diagram and this diagram shows us, is the movement of these gases based on the exertion of partial pressures in the system. So, time in the pulmonary capillaries. Not much time, right? How much? 0.75 what? Yeah, seconds. And then the percent of oxygen in millimeters of mercury. So by the time we pass through the pulmonary capillaries, what have we done to the pressure? Yep. Increased a little. And we've also held on to who? We're going to talk about that in a minute. Who are we holding on to when we leave? Who's oxygen going to hold on to when we leave? It goes from the atmosphere to who? It's kind of jumping on a ride. Hemoglobin. Very good. And how many seats does every hemoglobin have for me? It has 4 seats, doesn't it? You know what I'm talking about? Okay. So I have to jump on the ride in the alveoli. So we see in this diagram, which is a little confusion, is ventilation perfusion coupling, which basically talks about the movement of gases. And since it's a little confusing, I'm going to pass it. So ventilation perfusion coupling is a confusing way of saying gas exchange, moving to carrier molecules, and gas exchange on the other end when we talk about tissues, leaving those carrier molecules and going into the tissues. Remember, carbon dioxide is also going to want to hitch a ride on that molecule as well, and carbon dioxide is carried at the tissue level back to the respiratory system. They also give a little mention about the thickness and surface area of the respiratory membrane. And in a healthy lung, the respiratory membrane is only 0.5 to 1 micrometer thick. So that gas exchange is very efficient. What's going to happen if, for some reason, I get cells starting to form and thickening those respiratory areas? Hypertrophy of those regions. Am I going to have gas exchange happening? Not as easily as if my respiratory membrane was very thin. So they talk about certain homeostatic imbalances under this category as well. Too much fluid, too many cells. So give a quick read about that as well. And internal respiration now. Now we're talking about movement of gases where? When we talk about internal respiration, we're talking about gas exchange where? At the tissues. This is where we're going to drop it off. Now, one of the very important things we must remember is we have to get off the ride. Do you know what I mean? If chemistry does not allow me to get off the ride, and I'm talking about oxygen being carried by hemoglobin, gas exchange is not going to happen. It's not going to be efficient. So in this section of the book, they talk about something called an oxygen dissociation curve. Oxygen hemoglobin dissociation curve is what this one is. Basically, oxygen getting off and being distributed at the tissue level. Now, there's a bunch of different factors that are going to affect that. Temperature is going to affect that. Concentration is going to affect that. Give me some other ones. pH, huge factor in oxygen dissociation. If the pH is off, the chemistry of getting off the ride is not going to work. And we just talked about one. Yeah, those partial pressures. So I have to want to have incentive to get off the ride. I have to have a pressure difference in order for me to leave the hemoglobin molecule and go into the tissue level. So this is a hemoglobin saturation change as blood PO2, what's that? Partial pressure of oxygen changes. Notice that hemoglobin is almost completely saturated at a PO2 of 70 millimeters of mercury. So at 70 millimeters of mercury, most of the seats are full on the hemoglobin ride. Saturated. You see what I mean? Rapid loading and unloading of O2 to and from hemoglobin occurs at PO2 values in a steep proportion of the curve. Approximately 25% of hemoglobin bound to O2 is unloaded to resting tissues. Only 25%. So what's that mean? Do I unload all of it? Okay, so thus hemoglobin of venous blood from resting tissue is still about 75% saturated with oxygen after one round through the body. During exercise, PO2 can drop as low as 15 millimeters of mercury, causing an additional 50% unloading and leaving the hemoglobin only 25% saturated. So different situations will give us different proportions of unloading. This curve will change slightly. That's why when I stop breathing, I'm not going to die immediately. Why? Because I still have a bunch being carried around. Eventually, after more trips, those guys are going to get off the ride, but I still have time. I still have enough oxygen being carried by my hemoglobin molecules and in my blood plasma to be distributed to the tissue so they don't immediately start dying. And that was the basis for the development of the procedure of CPR. If I can get more oxygen into your blood, I can keep you going for longer, the pulmonary part. And if I can then pump that around your body, I can keep you going for longer. And that's saved countless numbers of people since its development to be able to do that. So That's an oxygen hemoglobin dissociation curve. So, influence of PO2 on hemoglobin saturation is important to know. Influence of other factors on hemoglobin saturation, and these are some of the different hemoglobin saturation curves. So, 70, what's the best temperature on this whole chart? Remember, we should have a peak. Yeah, which is the best one? Pick one on that one. Normal body temperature. Oh, I just gave you the answer. Yeah, 38 degrees what? Celsius, which is what? Well, 37 degrees Celsius, or 98.6 degrees Fahrenheit. That is the best saturation curve. Well, you're saying, well, no, Maria, 10 degrees is better. No. How come? You got to get off the ride. If you don't get off the ride, you're not doing my tissues any good. Are you going to get off the ride as easily at 10 degrees? No! You're going to hang on. That's not doing me any good in the tissues, is it? I have to let go. What about 43 degrees? I'm letting go too much. Yes? My molecules move faster at a higher temperature. It's kind of hard to, you ever get on one of those Ferris wheel things and somebody starts pushing it really, really hard? You're moving too fast? And sometimes kids like go flying off. Kind of like that. So 38 degrees is the optimal temperature for oxygen saturation of hemoglobin and for the ability to let go when I need to let go. How about over here? What's this? pH. What's optimal pH? Yeah, normal arterial carbon dioxide amounts, which equates to a normal pH of 7.4. 7.35 to 7.45 is your normal range, very tight range for pH. All right, what happens if the pH gets too acidic? I'm going to do what? Let go too fast. Is that good? No. You're going to drop me in places before I even get to the end of the ride. I'll be hanging out on the track somewhere. Is that going to do you any good? No. So, increased carbon dioxide. When do I get increased carbon dioxide? It's the opposite. Do you ever hyperventilate? What is that? You're letting go of too much carbon dioxide. That's the opposite. If you decrease carbon dioxide, what's going to happen to your pH? It's going to become too basic. What's going to happen to your oxygen? It's not going to let go when it's supposed to. So when you hyperventilate, you create a change in your pH because you get rid of too much carbon dioxide. You cause that pH to become more basic. Anybody ever hear of alkalosis? That's one of the reasons why you might go into respiratory alkalosis is if you hyperventilate. If you hypoventilate, you're not getting rid of enough carbon dioxide. You're increasing carbon dioxide. You're decreasing the pH. It's too acidic. We're going to talk about the reaction in a moment that creates that environment. So hypoventilation, not getting rid of enough carbon dioxide, can result in respiratory, what do you think we call this one? Acidosis. Hyperventilation, getting rid of too much carbon dioxide, can result in respiratory alkalosis. You with me? So, I gave you that quick explanation, so we could go through some of the descriptions under homeostatic imbalances. Hypoxia. What's hypoxia? Inadequate oxygen exchange at the tissue level. And that's messing with my oxygen saturation curve in some way, shape, or form. So poor oxygen delivery resulting in too few red blood cells, or from red blood cells that contain abnormal hemoglobin, or messing with the chemistry. Ischemic or stagnant hypoxia. Histotoxic hypoxia. Hypoxemic hypoxia. All relating in some way, shape, or form to either something wrong with the hemoglobin molecule or something wrong with all of these factors relating to gas exchange and the oxygen saturation curve. They also talk about something called carbon monoxide poisoning. Carbon monoxide is a ride hog. Anybody ever cut you when you're waiting in line at the Ferris wheel? That's carbon monoxide. And once they get on the ride, they're not getting off. You with me? So carbon monoxide, which is CO, not CO2, has a very strong affinity for the hemoglobin molecule. That's the fancy way of saying it. But what I'm saying is it's going to get on, and it's not getting off for a long time. So if that happens, what do you think is going to happen to gas exchange? Yeah, because the ride's full of carbon monoxide. So it sort of takes up the space that normal respiratory gases would normally use during exchange at the tissue level and in the respiratory system. So what's going to result from carbon monoxide poisoning? Yeah, by suffocation. According to the textbook, hemoglobin's affinity for carbon monoxide is 200 times greater than the affinity for oxygen or carbon dioxide. 200 times greater. It's like super gluing them to the seat. Yes? Carbon dioxide transport is what they talk about next. So understand what these mean. Understand what these oxygen saturation curves mean. Okay? How much do I have, and my affinity for dropping it off. That's important. If I can't drop it off, I'm in trouble. Okay, here's the confusing stuff. But it's not confusing. Don't let it intimidate you. This is the chemistry behind what's going on in the red blood cell, in plasma, that's going to help with gas exchange. So oxygen release and carbon dioxide pickup at the tissues is what this particular diagram is showing us. Now remember, at the tissue level, we have a partial pressure gradient that's going to kind of push carbon dioxide into the circulatory system for me, and it's going to push oxygen from the circulatory system into where? Into the tissues. So that partial pressure is helping with the push. The chemistry is going to help with who's holding on to who to bring where. Did I just confuse you? Good. So carbon dioxide is going to meet up with water, and this is in the, what's this big red thing? It's not an inner tube, it's a red blood cell, to create something with the help of carbonic anhydrase. What's H2CO3? It's carbonic acid. This chemistry, along with chlorine, is going to help with transport of carbon dioxide. Because carbon dioxide has a seat on the hemoglobin molecule, doesn't it? And when we're at the tissue levels, I dumped off oxygen, some of it anyway, so there's some empty seats on the ride for me. And this is the chemistry that's going to allow me to attach. The other thing that carbon dioxide, and actually most of the transport of carbon dioxide, is actually going to take place in the plasma. And the chemistry involved in that is the reaction that we see above. So plasma proteins, buffers, HCO3, a buffer, is going to help carry a lot of that carbon dioxide back to the circulatory system for gas exchange. So I have some chemistry going on here that's going to allow me a seat on the hemoglobin molecule as carbon dioxide. And then a lot of my seats on the ride, in this case, are actually going to be on plasma proteins in blood plasma. Yes? Okay. Where's this? So this is oxygen pickup and carbon dioxide release in the lungs. Again, partial pressure is going to play a role in giving a push to some of these molecules. Oxygen is going to diffuse out of the alveoli because of the pressure, partial pressure difference, that gradient we talked about before, and push it into the circulatory system. In this case, this reaction here is going to help me get a seat for oxygen on the hemoglobin molecule. We're going to work the other reaction backwards because we want to get rid of carbon dioxide at this point. So that carbonic acid is going to work backwards with, again, the help of carbonic anhydrase to produce carbon dioxide and water. So then the partial pressure difference can take that guy, push him off his seat, and throw him into where? The alveoli. Again, the buffer system reaction is going to work its way backwards as well. So we can then take that oxygen from this reaction and put it in the seat, the empty seat on my hemoglobin molecule. Seat or two. Again, we're talking normal circumstances. If I'm exercising really hard, I'm going to have more seats. Yes? Does that make sense? So understand the difference in the reactions. Just want to point out, when I add something to one side of a reaction, this is a chemistry thing, what's going to happen to it? And I want you to look at the reaction on page 834. Well, let me write it here for you. Got to pick my pen. So, I'm talking about this reaction here. CO2 plus H2O, and there's arrows like this. What kind of reaction is this? It's a reversible reaction. Leads to what? CO3, and then we'll get another reaction like this, and that leads to plus who? What's HCO3? It's a buffer. Okay, it's a buffer that's going to help pick up excess what? To make, and eventually that. Remember, these are reversible reactions. So if I add a lot of excess carbon dioxide to this mix, where's this reaction going to go? It's going to go this way and produce more what? Carbonic acid and more Remember we just talked about when I have too much carbon dioxide in my system, what happens to the pH? What happens to the pH? No, if I have too much carbon dioxide, it's going to decrease because I have an increase in yes? If I go the other way and pull out carbon dioxide, I don't have enough. Where's this reaction then going to go? That way. This is going to get taken up too much. I'm going to decrease the concentration of hydrogen ions, and what am I going to do to pH? I'm going to increase pH. Remember the brush off the cells. You ready? What was the definition of pH? Let's sit down for this one. Concentration of in a solution. A low pH is high concentration of hydrogen ions. A high pH is a low concentration of hydrogen ions. Remember when we talked, and this is like way brush off the brain cells, when we talked about hydrogen and hydroxide ion concentration in solution? Remember H2O can break down into hydrogen ions and OH ions. Holy moly. Okay, that's where this is all coming from. So these reactions are very important in helping to maintain blood pH and concentrations of ions in solution. So these respiratory gases are going to influence your body chemistry. So even though carbon dioxide is a waste gas, it's not like we can get rid of all the carbon dioxide in our body. Is that going to screw us up? Huge. Yes? We need to maintain a certain concentration of carbon dioxide in our body so we can keep this reaction flowing back and forth in a nice even rate. So we can help to maintain blood pH at a nice even rate. Okay? You with me? Not too confused? All right. So that's what they talk about under the Haldane effect, the amount of carbon dioxide transported in blood, and the influence of carbon dioxide on blood pH. So that was our discussion just now. Yes? Good. Read it over there so you can reinforce it in your mind. So who's going to control all of this wonderful stuff? First of all, how do I know what's going on in blood? What do you think is going to be a big key factor as far as respiratory gas concentration? What's going to kind of be the chemical cue, maybe, to the guys who are responsible for monitoring all this stuff? Those are the respiratory centers, medullary respiratory centers. Where do we find them? My favorite word, the medulla oblongata and the pons, yes? But what do you think the chemical change is going to be to help maybe speed up my respiratory rate or slow down my respiratory rate? Any clues? No, that's going to take me too much time. Who? pH! Yes! So if somebody is in respiratory alkalosis or acidosis, you're going to see a change in their respiratory rate, because that's kind of the cue that's going to help these guys to tell me how fast or how slow I need to breathe. And these are the guys that are going to send messages to the guys that are responsible for making that thoracic cavity smaller or larger. Diaphragm's a biggie, and of course my intercostal muscles. So the dorsal respiratory group The DRG, located dorsally near the root of cranial nerve, who? 9. And the ventral respiratory group, the VRG, are going to work together to help respiratory rate function properly so I can have the proper pH. So we have things that speed up respiratory rate, things that slow down respiratory rate. And they all are going to interact together to help regulate respiratory rate by regulating the muscles of the intercostals and the diaphragm. Yes? So, know a little bit about those guys. Some other cues that they get, some other information that they're going to get are going to be linked to other parts of the body. So neural and chemical influences on brainstem and respiratory centers are going to be coming from stretch receptors in the lung. Example, when I blow up a balloon too much, if I keep blowing it up and I don't stop, what's going to happen to the balloon? It's going to pop. Do we want that to happen here? So at a certain point, what do I have to do? Stop breathing in. Can you just keep breathing in forever and ever and ever? No, because these stretch receptors are going to say, stop breathing in. It's time to let those muscles relax. It's time to expire air. So stretch receptors are going to send negative information to this region to stop the inspiration process. Stop that signal telling my muscles to open my thoracic cavity. Also, irritant receptors in the carina region. If I get some schmutz down there, what am I going to do? I'm going to cough. So those also send negative information to that region. Stop the in. Now it's time for out. You with me? Receptors in muscles and joints. Why do they have any influence on my breathing rate? Aha! It's hermit time in the cafeteria, right? Remember that discussion? So those receptors, those little Golgi tendon bodies and all of those guys we talked about when we talked about muscles, they're also going to have an influence on my breathing rate. When they start moving really quick, they're going to send positive information because I have to start doing what? Increase my breathing rate so that I can get more efficient oxygen exchange. So when my muscles start moving a lot, they're going to send signals to increase breathing rate. Central chemoreceptors, if I have increased CO2, increased hydrogen ion concentration, what's going to happen? Increased CO2, what do I have to do with it? I have to get rid of it. So that's going to be signals. So peripheral chemoreceptors are going to monitor oxygen levels, things in the aorta. Remember, those are important because that's my first pass of all blood that's gone through the pulmonary circuit. So if the oxygen levels are too low, what do I need to do to my breathing rate? Speed it up a bit to increase oxygen levels in blood. So all of these guys are going to work together with respiratory centers in the pons and medulla, and of course my cerebral cortex, which is responsible for everything, is going to help control breathing rate with all of the signals we just mentioned. Yes? So chemical factors are going to influence, pressure factors are going to influence, muscle contractions are going to influence breathing rate. Hyperventilation, an increased rate of depth of breath. How can I stop you from hyperventilating? Because unfortunately, back to this guy. Oop, wait a minute. Back to this guy. When CO2 goes up, what's it going to signal your respiratory rate to do? It's going to signal it to increase, which defeats the purpose, right? So what do I need to do if I'm hyperventilating? What do I need to do? I need to get more carbon dioxide into my system because I'm breathing it out too fast. Yeah. Why do you think they do that? When you breathe out, you're breathing out a higher concentration of carbon dioxide. Are you breathing out oxygen too? Yeah, you are. But the concentration of carbon dioxide is going to be a little bit higher. So if I keep recirculating that air, I'm going to get a little bit more carbon dioxide in the air that I breathe. So that will help to increase carbon dioxide levels and get me back to normal. Okay? So my peripheral chemoreceptors are going to help with that. Okay. Arterial Pco2. What's that? Partial pressure that's being exerted by carbon dioxide in the arteries. An increased carbon dioxide partial pressure decreases pH in the brain's extracellular fluid. What's another name for that stuff called? Brain's extracellular fluid. Cerebrospinal fluid. So central chemoreceptors in the medulla respond to that, and then they're going to signal the medullary respiratory centers, the respiratory muscles, increased ventilation, more CO2 will be exhaled, and arterial PCO2, and of course pH, because of that whole carbonic anhydrase and carbonic acid chemical reaction, will return back to normal. So all of these guys are going to work together to help regulate blood pH and oxygen and carbon dioxide concentrations. So that's what they talk about under the influences of PCO2, the influences of arterial pH, and then the interactions between CO2 and PCO2. Again, look at those chemical reactions and see who moves in which direction with an increase in carbon dioxide or oxygen. Influences of higher brain centers, the hypothalamus is going to play a role in respiratory center modification, the cerebral cortex, pulmonary irritant reflexes, and of course the inflation reflex. So we see all of those guys kind of linked together with some of the cranial nerves. Cranial nerve, who is going to help with this whole thing? 9 and 10. So, respiratory adjustments. We have to change what's going on in the respiratory system when we exercise. That, I hope, at this point in your anatomy and physiology career is self-explanatory. Yes? So when we exercise, why do we need to change our respiratory rate? Because we need more oxygen to make more ATP to move those muscles, and the moving of those muscles is going to generate more what? CO2. With respect to the respiratory system, we're going to generate more carbon dioxide, which needs to be what? Expelled from the body. So that's why I need to influence my respiratory rate. I need to adjust my respiratory rate. Altitude. When you go to high elevations, do people live there? Yeah. Well, not really, really high, like on top of Mount Everest. But go to Colorado, people live there, right? When the boys go and play basketball there, sometimes they have a hard time. How come? Yeah, they're not used to the higher elevations. They're used to, like, down at good old disgusting Charles River level, So maybe that's a little bit of a plus on their side. The other teams has an advantage if they're used to that. So their oxygen saturation curves are a little bit different because of the difference in pressure in their atmosphere. So when I go to a higher pressure, higher above sea level, what happens to the partial pressure of the gases? Do they go higher or do they go lower? They go lower. So again, that's going to affect the gradients that I have to set up. So when you go up higher away from some people say the air is thinner. Is there less oxygen up there? No, it's just exerting a what? It's exerting a lower partial pressure. So it's going to take you a little bit more until your body becomes acclimated to the pressure changes to get used to creating that pressure gradient. So until your body acclimates intrapleurally to the pressure that you're in, you have to do what? Yeah, you have to breathe in a little bit deeper, or you feel like you're out of breath when you're going to higher elevations. And that's one of the reasons. Homeostatic imbalances of the respiratory system. I know I brought this up before, but of course it's worth a second mention. A group of diseases called chronic obstructive pulmonary diseases are what we are going to discuss under homeostatic imbalances of the respiratory system. And this kind of is an umbrella of many, many different things that can disrupt respiratory efficiency. Now, there's a list there of some of the relationships between the diseases in the respiratory system. And of course, again, I will point out what is first on the list. I will read it again. More than 80% of patients have a history of smoking who have these chronic obstructive pulmonary diseases. Now, that's not to say everybody, because there's still 20%. But I bet you in that 20%, we also have patients who were exposed to what? Secondhand smoke. Dyspnea, difficulty or labored breathing, often referred to as air hunger, coughing and frequent pulmonary infections, and most COPD victims develop respiratory failure, manifest as hypoventilation. Have you ever had a hard time breathing? Is that a good feeling? Absolutely not. It's terrible. So if you make a choice to eventually have trouble breathing, you might want to rethink that. Just throwing that out there too. So, emphysema. What's emphysema? Enlargement of the alveoli. So your balloons get too stretchy. You might have problems with gas exchange. Getting rid of too much, basically, when you should be holding on to some of it. Chronic bronchitis. Itis means what? Inflammation of bronchioles. And that can cause problems with resistance and compliance. Yes? And that can cause problems as well. Asthma. Kind of the same thing with respect to effects on airways. Tuberculosis. That's a nasty little what? It's called Mycobacterium tuberculosis. And it's very contagious, and it has an effect on lungs, compliance, and those bronchial tubes. Lung cancer. Abnormal cell formation. And because your lungs are extremely vascular, what is it about lung cancer that can be an issue? If I have a lot of blood vessels, I also have a lot of what? Yeah, well, who else is going to help me with the whole fluid balance thing besides the circulatory system? The lymphatic system, yes? And if I develop lung cancers, and they talk about squamous cell carcinoma and adenocarcinoma and small cell carcinomas, and those statistics are a little bit frightening as well. But what, again, I don't know if you're getting where I'm going. Well, no, don't smoke for sure. I mean, that hopefully. But those cancers can spread, and they can spread readily because of the fact that it's such vascular tissue. So they can hitch a ride in the lymphatic system and go set up house somewhere else.
Is it also on top of smoking, like oxidation, because there's a lot of oxygen exchange there too, so like free radicals?
Free radicals can also play a factor, but you're creating a lot of free radicals when you smoke.
Okay, now on top of that.
Yeah. Free radicals are nasty little molecules that damage other molecules. And free radical formation is something that you do every time you light up something. You burn anything, including throwing your steak on the grill. Not to scare you, but you create free radicals when you do that. And free radicals are nasty. What are the chemicals that can help you alleviate some of the damaging effects of free radicals? Oh, nutrition people. Antioxidants. So that's why they say eat your, red wine, that's right. And dark chocolate are high in antioxidants. But the other ones that you really should think about is a few blueberries or strawberries or some of the different, we call them super fruits because they're high in antioxidants, which are going to help neutralize the harming effects of free radical formation. So just a basic understanding of those diseases, developmental aspect of the respiratory system, don't worry about it, and we are done! Woo! Get your clickers out! It's clicker time! So any questions on the respiratory system before we clicker it up? All right, the first question. The blank zone includes the alveoli, while the blank zone includes the trachea. A. Wait a minute, where's my little... my little thingies. Wait a minute, I gotta get my pointer back. Get my pointer back. All right, there we go. So A is the conducting and respiratory, B is the transport and ventilation, C is the respiratory and conducting, and D is the ventilation and transport. Did everybody click? Who didn't click yet? So 34 is our number? All right, what's the answer? The answer is C. Remember, the respiratory zone is the alveoli, yes? Breathing air through the nose provides multiple functions. What function would be most impacted while breathing dry air? A, the warming of air. B, the delivery or delivering the air to the lungs. C, providing a resonance chamber for speech. Or D, humidifying the air. All right, 4, 3, what's the answer? It would be D, humidifying the air, getting moisture into it. So when we go down to gas exchange, we have plenty of moisture for gas exchange to take place. Everybody understand why moisture is important? So sometimes when you're breathing really dry air, what happens? Think about what you feel like physically. Sometimes you might cough, because you're getting more schmutz down into your lungs, basically. It's not being caught up by a lot of the mucous membranes, which need to stay moist. You might get really dry nasal passages. You might even start bleeding. Bloody noses. Anybody get those in the wintertime a lot? Try getting some moisture into your air, the air that you're breathing, because sometimes that might play a role. All right, people who have their adenoids removed to cut down on snoring, ooh, maybe that's what I can do to my husband, are having their what removed? A, nasal septum, B, I don't even know what that word is, cone, C, conche, or D, it's not that one, it's not B, or D, the pharyngeal tonsils. Five, four, three. What's the answer? D. Oh, you don't want to remove your nasal septum. Occasionally, food or liquid will go down the wrong pipe, initiating a cough reflex. Which structural barrier has been breached if this happens? A, the laryngopharynx, B, the uvula, C, the epiglottis, or D, the glottis? All right. Oops. Stop. The answer is C. All right, men tend to have deeper voices than women because their vocal cords are A, their vocal cords have a wider opening, B, are shorter and narrower, C, have a narrower opening, or D, are longer and thicker. Five, four. There we go. What's the answer? D. Remember we talked about a cello versus a violin? So if they're shorter, they're going to be higher pitched. If they're longer and thicker, they're going to be lower pitched. So D is the answer. The most violent coughing is initiated when foreign objects contact the what of the trachea? A, the carina, B, the trachealis muscle, C, the tracheal rings, or D, the mucosa? All right, what's the answer? Nobody? Oh! It's called the carina. Yes! And it's down, remember the trachea comes down and it branches? There's a little area of cells we talked about. Down in that area, it's called the carina. So, ooh, that was, now you won't get that wrong on a test. A is the answer for that one. All right. Who do we have next? This is a big, I hate putting big questions. Nah, I'm going to skip that one. I hate big questions because they're hard to. All right. This one. The pressure in the alveoli is known as A, the intrapulmonary pressure, B, the intrapleural pressure, C, the transpulmonary pressure, or D, the atmospheric pressure? Five, 4, 3, 2. Who's 34? Push a button. Did somebody leave? All right. We're down to 33. Well, somebody's batteries ran out, right? What's the answer? A is the answer. Okay, my friends, go away. We'll have a quiz on Thursday on Chapter 22. We will begin Chapter 23. Adios.